Hello everyone, good morning everyone, good morning. Good morning all. Good morning. Okay, so friends, in the yesterday class, we have we discussed the transposons, and there is a small error in the question that what we have discussed actually. Okay. Everyone please notice this particular question, friends. This is the question that what we have discussed. You remember this question, we all people has given the answer as the D. Okay, so it's not the D, friends. The answer will be here, it's a C. Okay, because friends, in case of a Drosophila, every student remember, in case of a Drosophila, there are two type of transposons are there. So one is the DNA transposon, another one is the retrotransposon, is it? So if you quickly look into the DNA transposon, what did I tell you? The DNA transposon example is the P1, whereas the example of the retrotransposon is the copia element. And friends, if you look into the humans, the line and these two are the type of the transposons which are present in the humans, which are the retrotransposons. Hence friends, the answer is not D, the answer is C. Please don't get confused. In the yesterday class we have given the wrong answer. Please highlight, Sumaya said that the answer is correct. Even later I have checked the video. We have given the answer as D. Answer is C, friends. Please don't forget. Copia elements is one of the retrotransposon in Drosophila. Lines and signs both are retrotransposons found in the human genome. When it comes to the friends P1 is the transposon which is present in the Drosophila, which is a DNA transposon. Okay friends, please don't get confused. Here the answer will be C. Okay, thank you Sumaya. Okay, so friends, in the yesterday class we have discussed about the, what we can say, the concept of transposon. So friends, now today we are going to discuss about the microbial physiology. I want every one of you to understand one very important point, that in this particular microbial physiology, friends, what kind of questions that we can expect actually. Every student please pay attention, friends. In this particular concept, friends, we can get very different type of questions. Okay, now the question is here, what kind of questions we can expect in this particular microbial physiology chapter. Okay friends, listen my words very carefully. Here we can get a different type of questions. Good morning everyone. Here you can get the questions in the form of matching. If you let's look into the 2022 February paper which is recently happened 5 months ago. Now everyone see here, we got this kind of a question, which is what a matching question. But what I am saying is, all the time we don't get the matching questions. Good morning everyone. We get the question which is a graphical. Okay, we can get the question experimental. We don't get most of the time any kind of analytical or factual statement based questions. We can get the graphical question, we can get the experimental based question, and friends, even we can get the numerical questions also. Okay, we can get the numerical questions also. But mostly what I can say, most of the times we can get the matching and graphical questions. If you miss the matching and graphical questions, friends, there is a great chance that we can get the experimental based questions as well as the numerical questions. Okay friends, this concept is a little small concept, but information is abundant amount of information. I am requesting all of you to understand entire information. It's a kind of a rigid topic, my friends. You have to understand with a great patience. Okay, now listen to me. Microbial physiology, what exactly we people has to study here? We have to study the growth, yield and characteristic of a microorganism. We have to understand the strategies, how exactly the cell division is happening, what are the various methods that has been followed during the process of cell division, and the most important, where you can get the statement related question. If you get the statement related questions, you can expect the statement related questions from this particular stress response mechanisms. Okay, now let's talk about this briefly. Focus friends, microbial physiology, what exactly you have to understand by the word physiology. Friends, when I use a word physiology, and when I use the word microbial physiology, most of the students will think that we have to study the microbiology. That is wrong. We are not studying the microbiology here. What we are studying, why they have given this in the cell biology in our CSIR syllabus, because we are not studying anything with respect to their microbiological properties alone. We are studying different characteristics of a microorganism. What do you mean by that? We are studying cell structure, we are studying growth factors, we are studying metabolism, we are studying genetic composition of the microorganisms. Many things we are studying, friends. That's the reason, every student please remember. See, you only tell me if we want to study the cell structure, we have to study the cell biology. Friends, please be patient for 20 minutes. Please listen to me very carefully. This is a rigid concept of patience checking chapter. So I want you to listen very calm and patiently. So in order to study the cell structure, we have to study the cell biology. In order to study the growth factors, what we have to study, friends? We have to study the microbiology. For the metabolism, we have to study the biochemistry. For their genetic composition, we have to study the genetics. So what is the point you people has to understand? Microbial physiology is an interrelated subject, where we are studying microbiology, cell biology, biochemistry and genetics with respect to what, friends? With respect to functioning of that particular bacterial cell. That is what exactly the microbial physiology is. Let me explain you briefly, friends. Growth, yield and characteristics. Now you tell me, when I ask you a definition, friends, how can you define the growth? When I ask you how can you define the growth, what you can define, if you consider a higher eukaryotic organisms like a human beings, how can you define, friends? We can define that increase in the number of cells, correct na? But now you only tell me, friends, when it comes to the bacteria, increasing in the number of cells we can say growth. But when it comes to the bacteria, friends, we all know that the bacteria is a single celled organism only, correct na, friends? What the bacteria is, single celled organism. Now the question is very clear, how can you tell me whether there is an increase in the number of the cells or not, when it is considering with the one bacteria? It is very difficult to say. So that's why everyone remember, instead of studying one, what we people will do is we will study the group. Every student remember, it is usually not convenient to investigate the growth and reproduction of individual microorganism. We can't study the individual microorganism because of their very small size. So it's a very simple point, my friends. Therefore, what the microbiologist will do, friends, whenever they are studying the growth, microbiologist always will study with respect to the total population number, because we can't study one. That's why we will study the population. Friends, when we are studying the population, we have to understand the growth curve very effectively. You all know very well, friends, when we are studying the growth curve. Now you have to ask me, sir, why you are saying the growth of a microorganism in the form of a curve? That is most important point. Friends, because the curve will explain us mathematical relationship with respect to the growth. That's why when we are measuring the growth of a microorganism population, we always measure in the form of a growth, which we can call microbial growth curve. So friends, this population growth curve having different phases. We can call it lag phase, we can call it log phase, we can call it stationary phase. There is another phase, friends, we can call it as the decline phase as well as the death phase. Once again, I am reminding to all of you, friends, it's a very rigid topic, lot of concepts to be remembered. I am requesting all of you only one thing: be patient for till end of the class. Then imagine 2 to 4 marks are in our hand, so be patient. Okay friends, when I use the word lag phase, I will use only one word, simple word I will use. Lag phase means preparatory phase. Anyway, I'll explain one by one. You remember when we are discussing the cell division, we understand very clearly that G1 phase is the preparatory phase for the S phase. What do you mean by that? In the S phase, DNA replication is happening. So in the S phase, DNA replication wants to happen. First, the cell has to prepare for the replication. So where the cell is getting prepared, friends, of course, cell is getting prepared in the G1 phase. Similarly, log phase is the phase where the growth is abundant. We can call it as the exponential phase. So friends, if the cell wants to grow exponentially, what is the statement you have to remember? First, the cell has to prepare itself. So in order to grow exponentially, the cell is getting prepared in the lag phase. That's why, friends, lag phase is said to be the preparatory phase. Kindly don't get confused with that particular point. Now, when I use a word stationary phase, I want you to remember always n equal to 0. In other words, I can say that birth equal to death. In other words, I can use the word cell division equal to cell death. Any way I can define it. Most of the students will say that stationary phase growth never occurs. That is wrong statement. And some students will think that stationary phase death also more. You have to remember, friends, in the stationary phase, always there is a equilibrium between the cell death and cell division. So you cannot say that in the stationary phase cell is dividing more, and you cannot say that in the stationary phase cell death is more. You should always say that there is a balance. There is a equilibrium between the cell division and cell death. Never forget that particular point. Next one is what, friends? Decline or death phase. When I use the word decline or death phase, you can't say that there is a growth. You can't say that there is a division. What you can say is there is a dramatic fall down. Remember, friends, there is a dramatic fall down in the number of microorganisms, which leads to the death of that particular organism. So let me explain you one by one phase very briefly and elaboratively. If you get the statement related questions, friends, you can get in terms of the statements with respect to the different phases. Otherwise, you will get the direct questions from the microorganisms itself. So pay attention. Please be patient. Now listen. Lag phase. Friends, I have taken, everyone remember, I have taken group of microorganisms. These are all a group of microorganisms. I have kept all these group of microorganism in the fresh culture medium. So friends, when I use a word fresh culture medium, what I have provided? I have provided everything. I have provided water, nutrients, I have provided amino acid, I have provided everything. And my friends, there is a most important point. There will be a no competition also. What, friends? There will be a no competition also. Now everyone remember, do you think growth will occur? Do you think even though I provide each and everything, do you think a growth will occur immediately? Never ever. What is the reason? Even though you provide all the suitable conditions for the microorganism to grow and develop, friends, they never grow very fast. What is the reason? The reason is, friends, that particular microorganism has to habituate, or it has to adapt to that particular culture. Once the microorganisms get habituated, or once the microorganisms get adapted to that particular culture medium, then only, friends, I can use the word, then only they start growing. Always remember. See, look into this line, every student. Focus, friends. When microorganisms are introduced into a fresh culture medium, so fresh culture medium means all are present, water, nutrients, minerals, and there is a no competition also. But what's the point we have highlighted here? Usually, friends, there is a no immediate increase in the cell number occurs. Hence, friends, therefore this period only we will be calling it as what, friends? Lag phase. Please do not get confused with this particular point. And now you can ask me a question, sir, if you only said that there is a no increasing in the cell number occurring, now can you tell me what is happening in the lag phase? Every student remember, cell division is not happening, friends, but the cell is preparing for the division. What I mean to say? I mean to say number never increases, but what is increasing is mass. It is a very important statement. Cell is not increasing its number, friends, but the cell is increasing its size. There is a great difference in the number and size. It can't increase its number because it's not yet prepared for the division. But it increases its mass. Why it is increasing its mass? Because it is preparing everything, whatever required for its growth and development. Which means, friends, the cell is going to synthesize a lot of growth factors. Cell is going to synthesize a lot of RNAs, lot of proteins and ATP, which is required to replicate that particular DNA. Always remember that particular point. So no increasing in the number, there will be increasing in the mass. And friends, here they have written one special statement. I want every student has to focus on this particular statement. What is the statement? Once the preparation is over, anyway, friends, the cell is entered into the log phase. Friends, when the cell is entered into the log phase, what is happening, friends? The growth is exponential. Yes or no? Which means that it is growing abundantly. Now, what I have done, whatever the cell, every student remember, whatever the cell that is growing exponentially or abundantly in the log or exponential phase, I have kept it back in the lag phase. Now what happens? Now you have to think and answer. Do you think when I introduced a particular cell which is already in the exponential phase, if I kept it back in the lag phase, do you think it will grow once again? Never ever. What's the statement you have to remember? The lag phase will be short. 90% of the cases, friends, lag phase is absent only. You only understand the cell is already prepared itself. After preparation only it entered into the exponential phase. Once again, if you keep back from the exponential phase to the preparatory phase, what will be the use? There will be no use. There is no need to prepare once again. It is already prepared. Hence, friends, the lag phase will be very short, or most of the cases, friends, it will be absent compulsory. Now, friends, the most important one, that is the exponential phase. Among all the phase, what is the most important phase? Undoubtedly exponential phase. What is happening? Friends, most of the students remember, when we use the word exponential phase, most of the students will think that the microorganisms grow abundantly and more in number in a irregular fashion. That statement is absolutely wrong statement, because, friends, the microorganisms never ever grow irregularly. The microorganisms never ever grow randomly, friends. the microorganisms always will show logarithmic fashion. What friends? They are always showing what friends? Logarithmic fashion. So what do you mean by the word logarithmic fashion? I can use the word 2 power n. In other words, let me define you exactly what do you mean by the word logarithmic fashion. Please remember friends, the microorganisms are dividing or are doubling in number at regular intervals. What do you mean by that friends? Which means at a particular time only the particular number of cells are getting divided. It's not like that at particular time a random number of cells are dividing. No, at a specific time, at a specific number of cells only getting divided. And one more important point, most of the students will think that if a particular cell from the lag or a preparatory phase, once it enter into the exponential phase, most of the students will think that it is growing exponentially all the time. That is also a wrong statement. You have to remember, when a microorganism wants to grow exponentially friends, there should be some conditions. What are the conditions? The genetic potential of that particular microorganism should be stronger, the nature of the medium should be stronger, and the conditions under which they are growing. Here the word conditions under which they are growing means friends, there should not be a competition. There should not be a competition. First point, nature of the medium means we have to provide all the suitable nutrients, water, minerals, amino acid for their growth. And even though you provided a good nutrient medium, even though there is a no competition, suppose example the microorganism having a less genetic potential, which means the microorganism do not have the ability to grow. You have to listen to me correctly. You have provided everything nice, you have provided good medium, there is a no competition, but the microorganism do not have the ability to grow and develop, then it cannot grow. So friends, in the exponential phase, if the microorganism wants to grow, there should be a genetic potential, nature of the medium, and there should not be a competition. Then only friends there will be a growth, that is exponential growth. And friends, always we can use the word what? Their rate of growth is, every student remember, constant in dividing and doubling in number at regular intervals, which we can call friends balanced growth. I want every student to look into this table. One second. Every student please look into the table. This is called as a logarithmic fashion. At time 0, the division number is also 0. The total cells are just one cell. But you see here 20 minutes, this is called division 1, cells are 2. 40 minutes, division 2, cells are 4. 60 minutes, division 3, cells are 8. You can see now friends, it is always following the 2 power n, and you can see here time period, it is always 20 minutes and division number is increasing episodically. We can use the word logarithmically. So what is the statement I can give you? In the exponential phase, the division number or the doubling number with respect to the time is equal, is balanced, is logarithmic. I want you to remember that particular point without fail. Now friends, look into this point. Now I have used a word that the exponential phase is possible whenever there is a good genetic potential, nature of the medium, as well as friends the conditions under which they are growing. This is only a very good condition. Now every student keep in your mind without fail, what I have done, I have disturbed the nutrient level. Otherwise, I have introduced the new species. Now, do you think the microorganism can grow exponentially? No, not at all. Do you think they can show the balanced growth? No, not at all. Friends, undoubtedly, they are going to show the unbalanced growth only. If a particular microorganism wants to grow exponentially all the time, there should not be competition and there is a limited amount of the, there will be adequate amount of the nutrients. Now we all know very well, friends, and we got the question also in the exam, that is the biphasic curve. Most of the time we can call it Diauxic growth curve. All the student remember what kind of question they can ask in the exam is in which of the following growth curves, what is the question they will ask, in which of the following growth curves multiple lag phases can be observed, means two lag phases will be observed only in the Diauxic growth curve. Now the question is very clear, when the bacteria can show the Diauxic growth curve. Now the question is, does the bacteria can show the Diauxic growth curve every single time? No, that is absolutely wrong statement. Diauxic growth curve cannot be shown every time. Diauxic growth curve can be shown whenever a bacteria introduced into a culture which is a mixture of the glucose also, as well as, friends, which is a mixture of the lactose also. Please don't get confused with this particular point. Now friends, I am a bacteria. What you people has done, I am a bacteria, you people provided me glucose and lactose, a mixture of the glucose and lactose. Now what I will do, being a bacteria, I never prefer the lactose. Initially I never prefer the lactose. Why I don't prefer the lactose initially? Because friends, lactose is a disaccharide. Being a bacteria, if I want to use the lactose, friends, first I have to break down the lactose. So I can't break it very easily. It is a time taking process, because friends, I have to synthesize multiple enzymes which are involved in the breakdown of lactose. So it's a greatly time taking process. So that's why instead of using the lactose, friends, I will always start using the glucose only. Never get confused with that particular point. So friends, whenever we are using the glucose, whenever I am a bacteria using the glucose, I don't require any enzymes. I can easily break it. Okay, so that's the reason. So my lag phase or preparatory phase for the utilization of the glucose is a very small, very small lag phase, because I don't require any enzymes. I'll just prepare myself and then I'll start consuming the glucose. But whatever the glucose that you have provided is a very minimal. So friends, what happen when I reach a particular stage, whatever the glucose that you have provided, everything is exhausted completely. But if I want to grow, if I want to survive, I require the food source compulsory. Anyway, the simple sugar like glucose is not available. Then obviously whatever the sugar is available, then I will start using that particular sugar. Now what is available here? Lactose is available, but the point is very clear, friends. I can't use the lactose directly. The reason behind that, first I have to synthesize an important enzyme called as beta galactosidase enzyme, which is involved in the breakdown of the glucose, lactose into glucose and galactose. So if I want to synthesize the beta galactosidase enzyme, friends, I can't synthesize this very immediately, friends. It takes a greater time. So my friends, every student, please remember, we have to wait for certain time. The bacteria has to wait for certain time to synthesize the enzyme beta galactosidase. Hence, what I can use a word, this is the second lag phase will be observed in the same culture. So that is what exactly, friends, we can call it as a Diauxic growth curve. Every student remember, Diauxic growth curve of E. coli grown with a mixture of glucose and lactose. Glucose is used first, then lactose. A short lag in the growth is present. See, everyone remember, we have used a word that a short lag. I am not using a word that the cells stop dividing. I am not using a word the cells stopped completely division. No, what I am saying is there is a short lag in their growth, very short lag. Why? Because it is preparing the enzymes required for the lactose use. That is what we can call it Diauxic growth curve. Friends, among all, as I told you, no, friends, if you get a statement related questions, there is only one way that you can expect the statement related questions from the stationary phase. I want you to listen to me very carefully. Now friends, every student commonly know that whatever the bacterium that is growing in the exponential phase, we read in the ecology also, it is going to give rise to which shape of the curve? It is going to give rise to the J-shaped growth curve. Correct now friends? It is going to give rise to the J-shaped growth curve. Now the question is very clear. We all people studied multiple times, especially in the ecology, none of the organism on the planet Earth never ever grow exponentially. Even, listen my words, none of the organism on the planet Earth never ever grow exponentially. Even slow growing organisms like humans, elephants, whales, none of the organism cannot grow exponentially. Once it reaches a particular point, immediately what happened friends? The growth will be stopped. The exponential growth will be stopped. What do we call it, that particular point? We can call it that particular point as a K. What do you mean by the word friends K? Every student remember, K means maximum carrying capacity that the environment can support. What friends? K is called as carrying capacity. Beyond that point, every student remember, beyond that point, environment cannot support for the growth and development of a microorganism. So after that, none of the organism never grow. So from the J-shaped, it is giving rise to the S-shaped growth curve. We can also call it as the sigmoid growth curve. Now, you have to remember a valid point, that is, now this is a very interesting point. Sir, how can you say, or based on what parameter you can conclude when a bacteria entering into the stationary phase? What is the proof that the bacteria is entering into the stationary phase only at this particular time? Yes, there are certain evidences are there that when the bacteria reaches a population size around 10 power 9 cells per ml, 10 power 9 cells per ml, then this is the point which indicates that the bacteria is going to enter into the stationary phase. Why? The reason is very simple. The nutrients are limited, and most of the students will get confused between the nutrients limited and nutrients depleted. Friends, please remember, this kind of statements only always makes us confused. When they use the word nutrients depleted means, friends, no nutrient is available. So what every student has to remember, undoubtedly bacteria will enter into the death phase. So, but when they use the word nutrients are limited, you should not think that bacteria entered into the S phase, bacteria entered into the stationary phase. Nutrients are present. Initially the percentage of nutrients are 100%. Now the nutrients percentage is what? 50%. The point is here, friends, nutrients are not absent, nutrients are not depleted, nutrients are present, but they are limited. So when the nutrients are limited, undoubtedly, friends, the growth is also limited. Very simple point. The growth is also limited. Now friends, this is the important statement every student remember. In the stationary phase, very unique statement it is. In the stationary phase, the total number of viable microorganisms remain constant. Friends, what do you mean by the word viable microorganism? Viable microorganism means which are reproductively active. Those microorganisms are remain constant. Remember that point. In one simple line, friends, I can tell you the stationary phase causes a balance between what? Between the cell division and cell death. Last and final and unique statement every student has to remember, friends. In the stationary phase, cell is not active. Cell is not active in terms of division. But what is the unique statement you people has to remember? The cell is active in terms of metabolism. Never ever forget this particular point. So they can give a statement that the cells are metabolically inactive in the stationary phase, but the cells are more active in the form of a division, which is absolutely wrong statement. Cells are inactive only in terms of division. The cell is not greatly dividing, but the cell is metabolically, friends, extremely active. Now these are all the various reasons. What is the what kind of part B question we can get? All of the following are the conditions allow the bacteria to enter into the stationary phase except. Now first I want you people to understand what are the various conditions that are allowing the bacteria to enter into the stationary phase. First one, see once again, remember the word. We have written the word nutrient limitation. We did not use the word nutrient depletion. Nutrients are present but they are limited, and especially aerobic bacteria, which they require the oxygen for continuously. Oxygen is available, my friends, but the oxygen is limited. Everything is getting limited. And the most unique statement I told you now, the bacteria population reaches K. What is K, friends? Carrying capacity. When a critical population level is reached, which we can call K. And another very important and unique statement, due to the accumulation of the toxic waste products. This is important because in the culture the toxic products are accumulating. If the toxic product is accumulating, how does the cell undergo the division? Never undergoes the division. I can use the word, this 4 are major reason for the cell to enter into the stationary phase. But friends, I told you just now, friends, we can expect the questions in the form of a statement. Now this is the most important slide. Whatever the every statement that I have written here, I am requesting every one of you to read every single statement without fail. Now you have to understand, when I said a word, when the bacteria enter into the stationary phase, they are not active in terms of division. Now most of the people will think that it is a negative experience for the bacteria, but friends, in contrast, the speciality of bacteria is that they can convert this negative situation into positive situation. What, friends? They are converting this negative situation into positive experience, which means, in one word I can tell you, bacteria are having the ability or capability to live in the stationary phase even years together. Understand? Do you think we human beings or higher animals, do you think they can survive for the long time in the starvation phase? They can't, because it is a negative experience for us. But for the bacteria, friends, it's a greatly positive experience. Why? What is the reason? Because, friends, when bacteria is, every student remember, when the bacteria is under the starvation phase, see here, there are many changes occur. What kind of changes? The changes in their gene expression and the changes in their physiological properties. I'll explain one by one change. Whatever the points that I'm highlighting, I want you people to remember all the points. Now the first and the major point every student has to remember, that, friends, whatever the bacteria that is under the starvation, every student remembered, bacteria is going to produce certain proteins. What do we call it, that particular proteins, friends? We call it them as the starvation proteins. Every student remember, bacteria producing what proteins, friends? Starvation proteins. Please don't get confused with that. You know what is the speciality of that particular starvation proteins? They will help the bacteria to become resistant. What, friends? To become resistant to, in a variety of ways. Let me explain you. First point is, in the starvation phase, bacteria releasing the starvation protein. And second unique characteristic is, friends, remember, the bacteria increase their peptidoglycan cross-linking and cell wall strength. You know, no, friends, the peptidoglycan of the bacteria made up of two important sugars called as NAG and NAM. You all people know very well, in the entire bacteria, which is the strongest structure? In the entire bacteria, friends, the strongest structure is the cell wall. Right? So what they are doing, they are increasing the cell wall strength. When they increase the cell wall strength, any kind of a enzyme or any kind of a protein, they cannot break the cell wall. That is the second one. And the third one, the bacteria start synthesizing a proteins called as DPS, DNA binding proteins, from the starved cells. So this DPS, friends, basically protect the DNA during the starvation condition. Please, friends, don't think that the DNA binding proteins can be synthesized all the time in the bacterial body. No, these DPS are getting synthesized in the bacterial body only when the bacteria is under the starvation state. And the fourth one, bacteria synthesizing abundant amount of the chaperones. We all know very well. We can also call it the chaperones as the heat shock proteins. What this particular chaperones or heat shock proteins are doing, friends, they are preventing, what they're preventing, they're preventing the protein denaturation and renature damaged protein. You all people know very well, if the protein is under renaturation, we can bring it to the normal activity. But you tell me, if the protein undergo the denaturation, can we bring it back? You can't bring it back. Protein which is in the renatured condition, you can bring it normal. Whichever is in the denatured, you can't bring it normal. So now you know what the chaperones will do, friends. 90% of the cases, chaperones will prevent the denaturation, means it never allow the protein to expose to the higher temperature. It never allow the disruption of tertiary structure of protein. What, friends? It never allow the disruption of the tertiary structure of the protein. If the protein is not biologically active, if the protein is in the renatured condition, then it will bring it to the normal condition. So friends, because of this four mechanism, ultimately what is happening, that is our ultimate goal. Ultimately, friends, the starved cell become extremely harder to kill and more resistant to starvation itself. Now every student remember, during the starvation condition, bacteria completely become resistant to the higher temperature. Bacteria become completely resistant to the oxidative as well as the osmotic stresses also. Even bacteria become greatly resistant to the toxic chemicals also. So that is the reason we can conclude a statement that some bacteria can survive the starvation for years together. So that's the reason we can give a valid statement that starvation can be a positive experience for the bacteria. The major 4 points you have to remember is, bacteria will release the starvation proteins, which makes the bacteria to become resistant. They increase the peptidoglycan synthesis and increase the cell wall strength. They synthesize the DPS protein and they synthesize the chaperones. These four are the unique mechanisms shown by the bacteria during that particular starvation conditions. So friends, there is no doubt bacteria can live for the very longer time in case of the stationary phase. Now the question is very clear. After the stationary phase, what is the phase? Death phase. I have very clearly told you, friends, in the stationary phase, everything is limited. But friends, when it comes to the death phase, everything is get depletion. There is a drastic fall down. What, friends? There is a drastic fall down in all the resources, nutrient deprivation and buildup of the toxic waste. Now friends, here comes one more interesting point. If you quickly recall, whenever we are studying the exponential phase, what did I tell to all of you? I told you that, friends, in the exponential phase, the growth is logarithmic, which means at a particular interval, once again listen, at a particular interval, the particular number of cells increasing. So when I use the word death phase, you have to remember that all the microorganisms never die at a time. That is also a wrong statement. What I would like to highlight, that death phase is also a logarithmic. What do you mean by that? Which means there is a constant proportion of cells dies every hour. What friends? There's a constant means equal number of cells are dying at a particular period of time. Now the question is very clear. What is the question? Now you can ask me, sir, how can you confirm? How can you confirm whether the bacteria has died or not? That's a good question. How can you confirm? See, every student remember, often the only way of deciding whether a bacterial cell is viable. Viable means, what did I use the word viable? Viable means whether it is reproductively active or not. What is the only way? Only way is by incubating them in the fresh medium. Now you only understand, I have taken the bacteria and I kept it in the fresh medium. When I kept it in the fresh medium, they are not at all growing, even they are not preparing for the division. Then what I can say, they are unable to reproduce. Then it is assumed to be dead. See what friends, what I have done, I have taken the bacterial cell and I have incubated in the fresh medium. Whenever it is not grown and reproduced, then it indicates that they are dead. So in one simple word, how can I define the word death? Every student remember, death is defined to be, everyone remember friends, death is defined to be irreversible loss of the ability to reproduce. Once again remember, irreversible loss of the ability to reproduce. When the microorganism is not reproducing, which indicates that the bacteria is dead. And what did I tell you friends? They are always usually follow the logarithmic fashion only. Please kindly do not get confused with that particular point. Okay. Okay, just 1 second. Yes. Okay, everyone answer this question number 21. Don't be hurry friends. Even though you know the answer, think and answer. Don't be hurry. Don't be hurry. B. Okay. Others. Okay. The question says that Diauxic pattern of biomass growth is associated with it. So we know that in the Diauxic growth curve there is a multiple lag phases. So P is right. So you can eliminate these two options. And sequential utilization of the multiple substrates. Correct na? First bacteria uses the glucose. Once the glucose is completed, then only bacteria go for the lactose. Otherwise it don't go for the lactose. Then Q is also right. Or they use the word simultaneous utilization, which means bacteria never ever uses glucose and lactose at a time. No, that's wrong statement. Absence of lag phase, wrong option. Hence the correct option will be the B. Good. Don't be hurry. Just think and answer. Which of the following events occur during the stationary phase for bacterial growth? Slowly, friends, slowly. Think it, take your time, take your time. Anyone? Slowly. I am giving you hint, I forgot to tell this particular line to all of you. I'm sorry. See friends, there will be certain morphological changes are taking place here. I want you to look into this. There is a certain morphological changes are happening, such as endospore formation. So I am just giving the hint that a spore can be formed. Friends, you have to answer this question with intelligence. Here, what is the difficulty in this? All the students has to look into this and answer my question first of all. Every student first answer my question, everyone. Everyone answer. Do you think is there a decrease in the peptidoglycan cross-linking or there is an increase in the peptidoglycan cross-linking? Everyone answer first. Is there is a decrease in the peptidoglycan or increase in the peptidoglycan? Everyone. Is it decrease or increase in the peptidoglycan cross-linking? Please respond everyone. Obviously friends, there will be an increase in the peptidoglycan. When you understand that there is increase in the peptidoglycan, very good. Option T is wrong. Hence you can eliminate these two. Very simple. Now the answer is in between A and C. 90% of the students has given the wrong answer. When did I say? You are not listening the lessons correctly, friends. You are not listening correctly. They are asking stationary phase. How many number of times I have told to you in the stationary phase what is happening? Every student remember. Every student remember. Here I have given a word that balance between the cell division and cell death. Always there is a balance is happening. Then how can you say that the rise in the cell number stops? How can you say? How can you say rise in the cell number stops? Just now I used a word, friends. There is always a balance between the cell division and cell death. So when the division is happening, how can you say that the number is stopping? How does the cell number will stop? Cell number never stops. So always, suppose example, 10 cell got divided, 10 cell got died. There is a balance is happening. You should not use the word there is a rise in the cell number stops. This is a wrong statement. Every student has chosen this statement. Spore formation. So if you come to know that this is a wrong statement, you can come to the option which is the answer C. Spore formation in gram-positive bacteria, right statement. Cell size increases, right. Growth rate of bacteria nearly equals to the death rate. Option yes only they have given in the form of option P. Rise in the cell number stops. That is a wrong statement. Always there is a balance occur. Hence QRS is the correct statements. Hence the correct answer will be C. Friends, everyone please respond. Is it clear or you want me to explain? Is it clear everyone? Please respond, please respond. Is it clear? Okay, perfect. I hope everyone understand without confusion. You can ask me if you have any kind of a confusion. Okay. So friends, in the Monday class, we will discuss the numerical part as well as the mathematics. Numerical part as well as the mathematical part we will discuss. Okay. We will do three models. We will do three models in terms of the generation time. See, in our CSIR, they never used to ask the questions from the generation time much. But the exams like ICMR, exams like GATE Life Sciences, they used to ask the questions from the generation time. but it is there, we have to do this compulsory, and especially we have to do the different type of bacteria and their representative organism. I will compulsory, I will discuss the numerical also, numerical as well as the graphical. But you know, from the last 3 years we are getting this kind of questions only, either graphical or the statement based questions, or are the this particular matching based questions. Okay, so friends, once again the class will be on Monday onwards, same time 3:30 p.m. only, without you know, the same timings. So this kind of questions they use it to ask more. Okay, we will discuss this also. If everyone finds the time, I am just requesting all of you, anyway I'll discuss in the classroom. If you find the time, please practice generation time numericals from this particular thing. Okay, anyway I will discuss 3 to 4 variations with 1 to 2 numericals, more than sufficient. We will solve 7 to 8 numericals and we will discuss the graphical questions. Beyond that level the question won't go at all. Okay. Okay friends, I'll upload this particular PDF in the app. Okay, Deepika, once you click on the crash course, you will get an option called as a live class and notes. Okay, notes, there you can, once you click on the notes, you can get it automatically. Okay friends, just 1 minute. By the next Friday, every student remember, next Friday, Saturday, Sunday, every student please remember, next Friday, Saturday, Sunday, I will upload chapter wise test. That is, I want every student, remember my words, on Friday I want you to take unit 1 and unit 2 chapter wise test. On Saturday I want you to take unit 3 and 4 chapter wise test. On Sunday I want you to take unit 5 and 6 chapter wise test. Then after 3 days once again I will upload the remaining. So Friday, Saturday, Sunday, every student has to take this particular things without fail. Okay, there will be a 45 questions are there, 25 questions from the part B and 20 questions from the part C. Without fail, please take the test. I will check each and every one result without fail. Okay, please update yourself by practicing that. Okay. Okay friends, thank you all. I'll see you in the Monday sharp 3:30 p.m. Thank you all of you. Please read well. Do not waste the time. Work more time. Okay. Bye bye. Thank you.